So looking at the synaptic area, we have the probability of excitatory postsynaptic potentials or inhibitory postsynaptic potentials. The excitatory simply mean we will try and depolarize the next membrane we come in contact with. We are promoting the action potential. Inhibitory, we are trying to hyperpolarize that next membrane or suppress the action potential. Okay? We can receive, a postsynaptic neuron can receive many different signals at once containing either excitatory or inhibitory. So neurotransmitters acting as the chemical messengers at the chemical synapses will cause either an excitatory or inhibitory response. The receptors have two structures, either ionotropic or metabotropic. Neurotransmitters' binding site and ion channel are part of the same protein, will be ionotropic. Metabotropic, neurotransmitter attached to one protein which open ion channels. So here's a lovely picture of those. Removal of neurotransmitters, we can either have diffusion, so neurotransmitters just leave the synaptic cleft through diffusion. Enzymes can cause breakdown of the neurotransmitter, or we can uptake those neurotransmitters back into cells. Summation of the synapses, so several presynaptic embulbs can release their neurotransmitters at about the same time, causing a combined effect, generating nerve impulses for summation. We have two different types, spatial or temporal. Spatial is going to be a buildup of neurotransmitters from multiple neurons for the presynaptic embulbs, causing that postsynaptic neuron to reach threshold. Or temporal, a buildup of neurotransmitters from the same embulb, releasing neurotransmitters at different times to try and reach threshold. So spatial summation, multiple neurons to one neuron, trying to cause threshold to be reached. So see here, some multiple stimuli to reach threshold. Temporal summation, we have one neuron continually releasing neurotransmitters until we can build up enough to cause threshold to be reached, creating an impulse. Now we can have multiple neurons attaching to a single neuron, sending either excitatory or inhibitory. Whichever one causes the most attachments of those excitatory or inhibitory is going to win out, meaning we will either have more excitatory connections, creating a continual action potential through that second neuron, or inhibitory will win out and we stop the action potential and will not send it. Okay, so neurotransmitters, the small molecule neurotransmitters, acetylcholine, this is what we see at the neuromuscular junction and quite a few other places. Amino acids, biogenetic amines, these are ones that we talk about a little. Neuroepinephrine and epinephrine, you can think of adrenaline, so we'll see these with the sympathetic system. Dopamine, we'll talk about in just a second and in the next chapter. Serotonin is kind of a feel-good neurotransmitter. ATP and other purines are excitatory. Nitric oxide is excitatory in the CNS and adrenal glands. We only form as needed. Carbon monoxide is formed as needed. Okay, so norepinephrine, again, this is kind of our adrenaline released from the adrenal glands. We are excitatory and causing a depolarizing effect associated with the sympathetic nervous system. Dopamine in the central nervous system we will see is either excitatory or inhibitory, but it is involved with Parkinson's disease. So if we do not create enough dopamine, this is where Parkinson's disease shows up. Serotonin, again in the central nervous system, it affects attention and emotional states. So if we do not create enough serotonin, this is where we may need antidepressants. Neuropeptides, we can serve as neurotransmitters and hormones. So these are just a few of the different types. These are their descriptions. Neural circuits, we have several different ways of connecting neurons. Simple series, one-to-one. Diverging, we can have one neuron and this will split into connecting with multiple neurons. Converging, multiple neurons condensing into one neuron. Reverberating, so we create loops which feed back on themselves, which is rather nice considering breathing has this reverberating connection. Parallel after discharge, so we have extensions from an axon gaining support from other neurons to connect to one neuron. We'll see pictures of these. So diverging, we are splitting the signal to spread that signal. Converging, we are condensing the signal. Reverberating, so we have connections into the main neuron that we're trying to reach, but we have a branch that says, why don't we keep this going? So we connect back to these other neurons that keep the signal moving on. So sort of a positive feedback loop, if you would like to think of it. Okay, so parallel after discharge, meaning one neuron has branches of its axon gaining support from other neurons to converge onto a single neuron. Some clinical notes. So regeneration and repair of nervous tissue, very difficult in the cerebrum or central nervous system in general. This is partially due to those astrocytes. They create scar tissue faster than the neurons can heal. But plasticity, this capability of change based on experience. So we see this a lot in the peripheral nervous system. So developing fine touch. We are learning, in other words. Regenerate, capability to replicate or repair, again, very limited in central nervous system, but in the peripheral nervous system, we see this a lot. Okay, so we have inhibitory influences from the neuroglia like astrocytes, and particularly the oligodendrocytes. So those oligodendrocytes creating that myelin sheath, they are not allowing for continual regeneration of an axon. Okay, absence of growth stimulating cues. So during fetal development, we are constantly growing the central nervous system, but as adults, we have what we're supposed to have. And again, rapid formation of scar tissue from astrocytes. Okay, this should be PNS. So in the PNS, Schwann cells help out quite a bit. Big thing is Wallerian degeneration. So what that means is the distal end of the neuron that has damage is going to degenerate and leave. And then the Schwann cells will form a tube-like structure to allow that axon to regenerate within it. And that is it.